Hello students, in our today's session we will be learning about X and Y linked gene disorders, the mechanism of sex determination and some genetic disorders which is covered in the chapter Inheritance and Variations. So let's get started. The first disorder that we are seeing today is color blindness. It is X-linked recessive disorder where person is unable to distinguish between red and green colors as both the colors appear gray. It is caused due to a recessive X-linked genes, capital X, small c, which prevents formation of color-sensitive cells, the cones in the retina of our eye. The genotype of the male and the female individuals can be represented as shown in the figure here. The homozygous recessive female having one capital X, small c, capital X, another c, and a hemizygous recessive male with one capital X, small c, and a capital Y are unable to distinguish between red and green colors, and hence they show the trait of color blindness. The frequency of colorblind women is much less as compared to a colorblind men. Dominant X-linked genes X capital C is necessary for the formation of color sensitive cells in the retina of the eye. So in this particular case where you have X and a capital C and X and a small c, the female in this case will not show the trait of color blindness but will be a carrier of that trait. The inheritance of color blindness can be studied in the following two types of marriages. The first case, marriage between a colorblind male and a female with normal vision. Have a look at the offsprings in the F1 generation. The sons will have normal vision and the daughters will be the carrier of the disease. And you see here the gamete formation is taking place, the gametes are getting separated and the crosses are shown of the chances of the two different types of gametes that will come together. So here you get 50% carrier daughters and 50% normal sons. The second case is a marriage between now one of the carrier daughters with a normal vision male. You have the genotypes here, you have the gametes formations, they separate and then there is equal chances of these gametes to come together, and then finally in the F1 generation we can see we have 25% normal female, 25% normal male, 25% carrier females and 25% colorblind male. Now from this example it is clear that X-linked recessive genes for color blindness is inherited from a colorblind father, this is the colorblind father, to his grandson through a carrier daughter. This type of inheritance is also called as criss-cross inheritance. Students, the next disorder is hemophilia, also called as bleeder's disease. Hemophilia is X-linked recessive disorder in which blood fails to clot or coagulates very slowly. The genes for normal clotting are dominant over the recessive genes for hemophilia. The person having recessive gene for hemophilia is deficient in clotting factors 8 or 9 in blood. Even minor injuries cause continuous bleeding. Hence, hemophilia is also called as bleeder's disease. The genotype of the male and the female individuals can be represented as follows. Here, capital X and capital H is the normal genes for the clotting factor and it is dominant, and here capital X and small h is the recessive gene for hemophilia. The recessive gene for hemophilia is located on the non-homologous regions of your X chromosome and there is no corresponding allele on the Y chromosomes to suppress its effect. So the men suffer from this disease. Women suffer only if both the X chromosome has the recessive gene or the alleles. If if the female has only one recessive gene and one dominant gene is present, then she is going to be a carrier for the disease of hemophilia. Like color blindness, hemophilia also shows crisscross inheritance. The inheritance of hemophilia can be studied with the help of the following example, where you have a marriage between a hemophilic male and a normal female. These are the genotypes. They separate out at the time of gamete formations, and you have the F1 generation. And just like you saw in color blindness, you have 50% carrier daughter and 50% normal son. Then the next marriage is between the carrier daughter and a normal vision male, where you'll have the gamete formations taking place, and you will have 55 out of the entire lot. You will have 25% normal females, 25% normal male, 25% carrier female, and 25% colorblind male. Again, in this case, you can see that the genes for hemophilia is transferred from the hemophilic father to his grandson through his carrier daughter, which shows the presence of crisscross inheritance. Sex determination. The mechanism by which sex is established is termed as sex determination. The term sex refers to sexual phenotype. In some species, both male and female reproductive organs are present in the same organism. It is described as a bisexual or a hermaphroditic or monoecious. On the other hand, some species in which the organism has either male or a female reproductive organs is said to be dioecious or unisexual. Humans are dioecious. A German biologist, Henking, in 1891, while studying spermatogenesis of squashbug, noted that 50% of the sperms received the unpaired chromosomes, while other 50% of the sperms did not receive it. Henking gave a name to these structures called as X bodies, but he could not explain its role in sex determination. Further investigation by other scientists led to conclusion that the X body of Henking was in fact a chromosome and gave the name X chromosome. Sex determination in human beings. The chromosomal mechanism of sex determination in human beings is XXXY type. In human beings, the nucleus of each somatic cell contains 46 chromosomes or 23 pairs. Out of these, 22 pairs are autonomous and 1 pair of sex chromosome. Human female has a pair of XX homomorphic sex chromosomes, while male has XY heteromorphic sex chromosomes. Thus, the genotype of the female is 44 autosomes plus XX and male is 44 autosomes plus XY. Human male is heterogametic and the female is homogametic. Have a look at the chart here. You have the male, you have the female. XY, XX. The gametes formation, you will have XY, X in one, Y in the another one, and two similar types of gametes, that is X, which clarifies the statement that the male is heterogametic and the female is homogametic. Yeah. The F1 generation now, if the sperm containing X chromosome fertilizes the ovum containing X, then the zygote will be formed and will grow into a female child. Here, if the sperm containing Y chromosome fertilizes the egg, then the diploid zygote will grow as a male child. This indicates that the sex of the child depends on the type of the sperm fertilizing the eggs, and hence it is the father who is responsible for the determination of the sex of the child and not the mother. Due to lack of knowledge, women are often blamed for giving birth to a female child. The sex determination in birds. In birds, the chromosomal mechanism of sex determination is ZWZZ type. In this type, the females are heterogametic and produce two types of eggs. 50% eggs carry Z chromosome while 50% eggs carry W chromosomes. have a look at this here. So the female has ZW, 50% will carry Z chromosome, 50% will carry W chromosomes. Male are homogametic and produced only one type of sperms. Each sperm contains the Z chromosome here. Thus, the sex of the individual depends upon the kind of egg or the ovum that fertilizes the sperm. Sex determination in honeybees. In honeybees, the chromosomal mechanism of sex determination is haploid-diploid type. In this type, the sex of the individual is determined by the number of the set of chromosomes received and not by the type of the chromosomes. Female here are diploid 2n32 and the male is haploid n16. The female produces haploid eggs by meiosis, that is N16 and the male produces a haploid sperm directly by mitosis. So there is no equational division happening in the male. If the egg is fertilized by the sperm, 2n is restored, then you have a queen or a worker and an unfertilized egg develops into a haploid male, that is N16, which is called as drone by ways of parthenogenesis. Parthenogenesis is a process where you have the development of an organism without the process of fertilization taking place. Genetic disorders. They are broadly classified into two categories as Mendelian disorders and chromosomal disorders. Mendelian disorders are mainly caused due to alteration or mutation in genes, example thalassemia, and on the other hand, the chromosomal disorders are caused due to absence or excess of one or more chromosomes or their abnormal arrangement. We will see a few disorders of both the categories. The first one is a Mendelian disorder, that is thalassemia. Thalassemia is an autosomal inherent recessive disease. Hemoglobin molecule is made up of 4 polypeptide chains, 2 alpha A chains and 2 beta B chains. The synthesis of alpha chain are controlled by two closely linked genes, HBA1 and HBA2 on chromosome 16, while the synthesis of beta chain is controlled by a single gene HBB on chromosome 11. Depending upon which chain of hemoglobin is affected, thalassemia is classified as alpha thalassemia or beta thalassemia. Thus, it is caused due to a deletion or mutation of a gene which codes for either the alpha A chain or the beta, that is the B globulin chains, that results in abnormal synthesis of hemoglobin. Now, what are the symptoms of this disease? You have anemia, pale yellow skin, change in the size and shape of your RBCs, slow growth and development, dark urine, etc. Massive blood transfusions are needed to these patients. Thalassemia differs from sickle cell anemia. The former is a quantitative problem of synthesizing few globulin molecules, while the later is a qualitative problem of synthesizing an incorrect functional globulin. To show you the difference between anemia and thalassemia, I've put up these two images. You can see here in anemia, your RBCs are less in number, but in case of thalassemia, the RBCs, the red blood cells, are malformed. They are not formed properly and so they are not functional in the patient. Students, now let's have a look at some chromosomal disorders. The first one is Down syndrome, also called as 21 trisomy. Now, this syndrome is caused due to an extra copy of only the chromosome number 21. It shows presence of 3 copies of 21 chromosomes instead of an homologous pair. So, the individual has 47 chromosomes instead of the normal number 46. 21 trisomy occurs due to non-disjunctions or failure of separations of chromosomes during gamete formations. This normally occurs in mothers who are over 45 years old. I've put a karyotype of the Down syndrome. You can see all the 23 pairs of chromosomes are put up, and you can observe that the 21st chromosome, instead of 2 pairs, you have 3, which gives rise to this disorder called as Down syndrome. Some of the symptoms that the patients show is mild or moderate mental retardations, poor skeletal development, small head, ears, small mouth. Face is typically flat, rounded with flat nose, open mouth, protruding tongue, eyes slant up and they are slightly moved out. So these are some of the symptoms that we commonly see. Students, the next sex chromosomal disorders are Turner syndrome and Klinefelter's syndrome. In Turner syndrome, you have one less X chromosome, and in Klinefelter syndrome, you have one more X chromosome. You can have